Good morning and welcome to Embryology. So this would be our concept. For next 2 days, we'll be learning embryology literally by drawing. Every step we'll draw, and I wish that you also parallelly keep on drawing the things, right? And as I said, keep 3 or 4 different colored pen or pencils and blank sheets. That's it. And all the way today we'll be starting from fertilization, right? In fact, we'll be starting with how the ovum is matured, right? Literally, we'll start from that early stage all the way till the formation of trigerminal layer, then the notochord, then the neural tube, neural crest, what really forms from that. That is till the point when the folding starts, right? That folding part will take tomorrow. So every concept, that is from where exactly the peripheral nervous system develops, from where exactly the retina develops, right? All these things, they will be covered into complete detail, so that by tomorrow you'll be knowing very crystal clear that which part develops from where. You'll be knowing various parts of the brain, and then from the next week onwards we'll start taking individually every part, and obviously the clinical aspect and the radiological aspect, right? So that's how things would be learnt. The whole idea for drawing is, when we really draw, we understand, right? And it makes our concepts very clear. You need not be artistic, but you need to be scientifically correct, right? So that's the only expectation. It really doesn't matter if you draw some of the cells like this, or if it becomes like this, it's okay, right? No, tomorrow though we'll be going for embryology. Right now we'll not be taking the test because this is very important concept. Let it finish, then we'll plan for the test. Okay, so let's start with our topic. Starting from that, this is the uterus, right? And then here is the fallopian tube, and that's the fimbrial end. And let's take a cut section, right? So let's take a cut section, and that's the fimbrial end, and here is the ovary, right? So this is the uterus. So we know that the ovary, from ovary at a regular interval every month, the ovum would be released. That ovum which would be released, it would be captured by this fimbrial end. So this is what is called as the fimbrial end, correct? Fimbrial end of fallopian tube. And that's the ovum, and it will travel all the way to the fallopian tube, right? This portion, right? This is, it is opening into the fundus, right? Fundus of the uterus. So that one is fundus. Few important thing about this. This is one of the important ligament. That is what is called as the proper ligament. This is what is called as the proper ligament of ovary, right? Proper ligament of ovary, which is very important for the support. And the entire thing is the fallopian tube. In fallopian tube, there would be, let's change the color. There would be one part which would be the intrauterine part, right? The part which is inside the, or you can just say uterine part, right? Uterine part. Then there is a junction which is called as the isthmus, right? That is what is called as the isthmus. And then the all the way up to the end, this one is what is called as the ampulla, right? And finally, this portion which is called as the infundibulum, right? Infundibulum. And finally, the fimbrial end. So that's how the ovum, it starts traveling and it reaches to that point. Okay, the fertilization, right? The fertilization, it actually occurs before it even reaches to the, when it reaches to the, say, the fundus. Those those sperms, they travel all the way, right? And somewhere over here, the fertilization occurs. That's where the ovum and the sperm meets. This is the reason of ectopic pregnancy. Ectopic pregnancy, that is sometimes when there is tubal pregnancy. Tubal pregnancy means the fertilization occurs over here in the fallopian tube, and then it gets embedded over there only, and then it becomes a major emergency, right? Because the tube can rupture very easily. So that's important to know, that this is the site of fertilization. Now let's go back even before that, right? This is an infant, a female infant. So in infant ovary, everything is in the form of, right? So these are the cells, right? These are all superficial epithelial cells, and then inside, these are what is called as the, you must know, primordial follicles, right? They are called as the primordial follicles. So right from the birth, female is carrying these ovums, primordial follicles. These are not matured, and they will remain there into this state till she is adult. So that's where the two-part tunica albuginea and the cortex. So initially this portion, which is called as tunica albuginea, and in the cortex part there are these primordial follicles. So these primordial follicles, in which there is, that's what is called as the primary ova, right? So that is lying, and it stays silently. Yeah, and it stays silently over there, right? As the follicles develop, right? That is, as she becomes adult, as she is adult, so there would be formation of the mature follicle, right? So then this ovum, right, which is surrounded by, right, which is surrounded by what is called as zona pellucida, right? Zona pellucida. So it is this zona pellucida which would be broken. That would be that when we'll be reaching to the stage of, uh, say, when it will fertilize, and then it will be embedded, right? After embedding. So at that point, zona pellucida will be coming into picture, because it has to, before hatching, it has to break this, and then it has to enter, right? Then rest, we are not going into that detail of the entire menstrual cycle, etc. But we just say that that's the point. That's the point when that we have got this ovum, which is now ready, and it is ready to release, right? So similarly over here, the cells which will be present over here, they are called as the corona radiata, and then cumulus euphoricus, right? Then there is this would be the whole follicular cavity, right? This would be the follicular cavity, right? Follicular cavity. And when the ovum is released, right, you know that it is called as the corpus luteum, rest of the thing which is left out, right? That is what is called as the corpus luteum, and then it becomes corpus albicans, etc. But the whole idea is, now we are talking about mature ovum, the mature ovum, which is into, right now it is in ovary, right? And it is about to get released. In ovary, there would be even other ovums. So there, some of them, they will be remaining as primary ova, that is the primordial follicles, right? Because everyone will not be getting matured in a single shot. No, there'll be few will be in the form of primordial follicles. Then few will develop and they will become the primary follicle, right? So primordial follicle to primary, right? Primordial follicle to primary follicle to secondary follicle, right? These are all stages. Secondary follicle, and then that secondary follicle, which is also called as the antral, and it will become the mature ovum, and that mature ovum which will be released, it is released alternately by one of the ovary, right side, left side, every month, and then it will be coming into the, it will be picked up by the those fimbrial ends. What is left out is corpus luteum, right? Okay, so we have now reached to that stage where we are ready to talk about the fertilization process. Acha, one important thing from the clinical aspect, right? From the clinical aspect, there are two concepts. One, when it is the date which is calculated by clinicians, right, or the gynecologist, and the date which is calculated by embryologist. Now, is there any difference? Yeah, there is a big difference. Today we'll be talking in the term of as if we are embryologist, because we'll be watching every stage. So when we say 24 days or when we say 30 days, right? So it's like how we are calculating. Let's draw it like this. This is LMP, last menstrual period, right? That is the first day of bleeding, first day of last cycle's bleeding. Now we know that roughly about 2 weeks, 14 days is the point when the ovum is released, right? Mature ovum is released and it is fertilized. So we'll just put it over here 2. That is 2 weeks. 2 weeks. And the fertilization occurred over here. Embryologists would start considering that the real process would start from this point onwards. Makes sense, right? It makes sense. So he will be considering this timeline. So when it comes to clinicians, when it comes to gynec, obstetrician, they will say, okay, no, we'll consider from the last menstrual period. Right? So they will be taking history and when was the last cycle, first date, and then they will calculate it in a way that 3 to 6 days that was the menstrual phase, right? And then starts the proliferative phase, right? And that proliferative phase which varies from 10 to 12 days. But one thing is very definite, that just before 14 days of the next menstrual cycle, that is the point of ovulation, right? I hope I'm clear, right? So if this is the whole cycle, let's say the cycle is of 28 days, so then calculate these 14 days, right? Before the commencement of the next cycle, this is where the ovulation would occur. That is when the mature ovum would be released. Let's say if the cycle is of 30 days, right? So over here it would be the 14th day. But if the cycle is of 30 days, so calculate from here, because this portion is fixed 14 days. This portion can vary, right? It can vary. There are various effects, right? Psychological stress, right? All those factors. So we'll discuss it at when we'll be talking about the gynec and obstetric part. But today we'll say that this phase is fixed. So that's why from the last menstrual cycle they just calculate it. Right? That's why they say okay, whether the periods were regular, irregular, what was the average duration, etc. So it makes sense over here. So they will start counting over here, and from that point onwards, that is the 40th week. This is for EDD, expected date of delivery, right? But from the embryology perspective, what he would say, 38. He would say 38. 38 weeks are needed. So that's why clinicians, they will be moving always two weeks ahead as compared to embryologist, right? Will it make any difference when you go for ultrasound, when you do ultrasound, right? When you are doing ultrasound, you are actually watching. So today we'll actually draw the notochord. We'll see that how the notochord is formed, how the neural tube. So when you are doing ultrasound, so you'll be able to watch the structures. When you watch the structures, you decipher that okay, this must be the age, right? But remember, if you are gynec, if you are obstetrician, so then there would be the gap of that 2 weeks. So that's the difference between embryologist or the radiologist. They will be watching because they will be evaluating what's the age as per their results on the screen. So radiologist, embryologist, they will consider this timeline. Clinicians, they will be considering this timeline. just to make it very clear that there will be the gap of 2 weeks, right? A very simple calculation, if it is to be calculated from the last menstrual period, right? Add 9 months, 9 months plus 7 days. Whatever comes, right? Whatever comes, that would be the expected date of delivery, EDD. Why this adjustment? Because on an average, it works in the form of a lunar months. So right, 30 days, and then adding those 7 days. So it takes care of those that some days are like 28 days month, some are 30, some are 31. So when we add this thing, it comes to almost near perfect prediction of EDD, expected date of delivery, right? This is a rule which is followed clinically. So that's the difference between the embryologist and the clinician's part. Now we start with, we just zoom this process, and this is right now we are into fallopian tube. Okay, so here the ovum is discharged. This is a mature ovum, right? This is the mature ovum. That's the zona pellucida, right? And it's discharged, picked up by the fimbrial end. Let's see the color. Picked up by the fimbrial end, and now it is over here, fertilized, right? So it's fertilized. So all the processes, right? That is the acrosome comes, then there is capacitation, rest of the chromosome, rest of the sperms, they are not allowed to enter, etc. But basically, this is now fertilized, right? When it is fertilized, moves to next level, right? This is what is called as the two cell stage, right? Two cell stage. This occurs in about 30 hours of fertilization. 30 hours of fertilization. Uh, you must have heard about that there are pills available which are, that is, till this fertilized ovum, if it is not embedded, right, it is possible just with the help of pill to prevent the embedding into the uterine wall, right? After that, things become difficult, right? So over here, you'll see that entire timeline, right? You'll see that entire timeline. So here is this two cell stage, 30 hours, right? It moves, and over here, if you really draw, it would be something like this, two cell stage, right? Two cell stage, two cell stage. After two cell stage, there would be mitotic division, and it will move into the four cell stage. So this is four cell stage. How much time? This is four cell stage. Four cell taking 40 hours, right? So after about, say this is fertilization, 0 hours, right? 0 hours, and 30 hours, two cell stage, 40 hours, four cell stage, and then it multiplies, right? It multiplies. And over here, right, it would multiply from 8 cell to 12 cell. By, in some books it is like 8 cell, or sometimes it is 12 cells, but now when it occurs about 12 cells, we call it early morula, right? This is what is called as the early morula, right? Early morula. So in this early morula, which is 8 cells, so easy to remember, 80 hours, okay? 80 hours, and then it grows, and now this is, I'll not draw all of them. Now this is about, say, 32 cells, right? 32 cells, and this is what is called as the advanced morula, right? So we call this thing as advanced morula, advanced morula. So this advanced morula, as it progresses, now there will be some major change, right? There will be some major change, and say over here 80 hours means almost, say, 3 days, more than 3 days, three. So over here, this would be, now we'll be talking about in days, right? From this point onwards, we'll be talking about days. So this is like 4 days. Done. So we have reached to this advanced morula, 4 days. Still, it is into the fallopian tube, right? From this point, let's move on to, now we are drawing it over here, but it is still, okay, well, let's draw it on another page, right? Still, things are like we are into this, okay? This is the fallopian tube, and this one is the uterus. So over here, that's the advanced morula. Drawing it slightly bigger now. Now there is a formation of a cavity, and it is called as the blastocele, right? There is formation of a cavity. So now those cells, they start moving towards one pole. So those cells, right? Now let's name these cells, because now is the right time to name these cells, and we make a change in the color. So these cells, these are the cells. They move on to the one pole, and these cells which are over there, they are called as the embryoblast, right? Or the inner cell mass, right? Or the inner cell mass. And this is the cavity which is formed, right? That is what is called as the blastocele. This is the hallmark, blastocele. And now this process will be called as this whole thing will be called as the blastocyst. This portion, that will be occurring after 1 day, that is about 5 days, right? 5 days. 5 days. Now is the point where it will start going for the implantation, right? So from here, it will go into the stage of implantation. So when it has to really enter into the uterine wall. So about 6 days, right? 6.5 days, 6 to 6.5 days. And that's what is called as the early implantation, right? The initial stage of implantation. Very interesting thing happens at this point of time. Now think it like this. This is the uterus, right? That's the endometrium, right? So we are just taking one side. So that's the endometrium, right? Endometrium. There has to be, this is compulsory, right? There has to be the proper alignment. So these cells which are over here, right? What we call as the embryoblast, or what we call as the inner cell mass, right? This one. By the way, these cells which are outside, they are called as the trophoblast, right? Trophoblast. Now these trophoblasts, they release what's called as the HCG, human chorionic gonadotrophin. So in all the prac test, right, it detects the level of HCG, right? Human chorionic gonadotrophin. So that is tested, right? So now, but this alignment is vital. So in case, because see, this is like a round structure, right? It is not necessary that it would be exactly alignment. This head, right? If we consider this thing as head, or the inner cell mass, or the embryoblast, so that embryoblast, it might be rotated. So no, it will keep on rotating till it is in proper alignment, and that is where it will start with the implantation, right? So this is one very important point. Yeah, HCG is the indicator of pregnancy, because that's the point when say things have developed to this much level. And because now nowadays the sensitivity of that test is very high, so that's why it is very precise. Okay, moving on to the implantation part. Now see, for the implantation, Now today what we'll do is that parallelly we'll keep on discussing even the abnormalities also, right? But from the perspective of a nervous system, right? CNS. So in the implantation, technically speaking, there are four stages, right? The stage one, in the stage one, what's called as the hatching, right? Hatching. In hatching, it is the blastocyst which will come out of the zona pellucida. That is the first thing. So that blastocyst which will come out of the zona pellucida. Zona pellucida, that was the outer layer which was covering it, right? All the way it got carried. Second stage would be, this is what is called as the apposition. Apposition. This is the stage what I was talking about. That is in this, that this trophoblast, right, that will come in contact with the endometrium, right? So we were telling about that these are the outer cells, right? These are the outer cells, and then this was the inner cell mass, right? Inner cell mass. So when this trophoblast, this one, right, that is the trophoblast, when it comes in contact with endometrium of the uterus, right, this is the stage of apposition. But then third stage, which is called as the adhesion. This adhesion won't occur. Adhesion occurs only when the alignment is proper, right? When the alignment is proper. That means when the head, right? This we are writing this head just for our convenience, right? So when the head, because it won't happen if it is reversed. So this portion, right? This portion will never be embedded into the endometrium. So a position, and then it will start rotating till the head is aligned with endometrium, right? So that is the adhesion. And once that is aligned all the way, it moves on to the fourth stage, and that fourth stage is invasion, right? In invasion, it entry into endometrium, right? Entry into endometrium. So now journey starts from this point, that because from this point things will be completely rapid, right? So let's draw it, right? So that we are properly oriented. Let's say this is day 7, right? Because 6.5 days that implantation starts, and by day 7 it is inside, right? So if we say that, okay, this is like uterine epithelium, right? Uterus, this is epithelium or the endometrium. That's the endometrium. And the whole cell, right, it is now inside. So let's draw it like this. Fine. So all these cells, they are, we draw this much, right? They are now called as the cytotrophoblast, right? Cytotrophoblast. There will be two types. One will be the cytotrophoblast, and second will be syncytiotrophoblast, right? Syncytiotrophoblast, they will be surrounding this. They will be surrounding this, because now the environment is getting created, because then the placenta is to be created, increased blood vessels, right? So that this would be the syncytiotrophoblast. Trophoblast. Difference between both of them, very subtle, very clear difference. This is mononucleated, right? Mononucleated. That is cytotrophoblast. And syncytiotrophoblast, they are multinucleated. And syncytiotrophoblast, they will be outside, right? This we said it produces HCG, right, in large amount. Now inside, Now the game starts with the type of cells. So we'll draw it like this. Say this is one group of cell, and then there is another group of cell which is like this. So that inner cell mass, right? That inner cell mass, which was like same type of cells, but now, right? So it is like this. What are these? This one, writing in capital, it is called as the epiblast. Epiblast. And this one, that is called as the hypoblast. Hypoblast. Hypoblast, that would be extra embryonic. Right? Just remember this part, that this is, right? That one would be extra embryonic. And this is the epiblast, which will lead to the formation of amniotic cavity. We'll see this, right? So far, and yeah, this is uterine epithelium. Okay, this one. So now it is entering inside. Two layers already ready, right? Same thing now changes to this. Let's say this is day 8. Now in day 8 only one major change. So now onwards, which we'll draw like this, and these are the surrounding cells, right? So we'll draw them a bit quickly. So these are, and yeah, every cell will be having their right nucleus inside, but we are not drawing it. And then over here, this is that uterine epithelium. So you know this part, right? So this is the cytotrophoblast. We'll write it like this, and this is the uterine epithelium. See, it is now entering deeper, right? This is all internal structure, right? This is outside. This is outside, right? And the direction of movement is that is going in. And which color we have used for that? Okay, yellow, and we'll take a different color. Take it. Huh. So that let's take yellow. So that one, right? So this is one layer, right? Which we called it as hypoblast, right? So over here, this is hypoblast. And another one, which was above. Now see, these will be the columnar cells, right? These will be the columnar cells, right? Columnar cells. And then there would be development of one cavity. Right? The cells which are over here, they are now called as the amnioblast. Amnioblast cells. And this is the cavity which is formed over here is the amniotic cavity. Amniotic cavity. Right? So we have now with us this portion, this, and that's called as the, this one is called as the two layer, right? Two layer. So that is also called as the bilaminar. So bi means two, lamina means layer. So it is bilaminar disc, right? So this whole thing would look like a disc, right? This one, this entire thing is like a disc. Fine. So this implantation continues, right? So by day 9, day 8, day 9, this it reaches to this level. What about the other part? It moves to, now we'll just draw it like this bigger one, right? So considering that out is all cytotrophoblast, right? We are not drawing it now. So it is all cytotrophoblast. Then we have, we are using yellow and this. Okay. So we have one layer, right over here, right, and then we had those. So this was our amniotic cavity, right? There would be development of over here, and that is called as the primary yolk sac, right? So this would be primary yolk sac. This will keep on enlarging, right? So this is day 9, right? Day 9. What we are trying to understand is will reach to the level of trilaminar disc, and then the game starts for the notochord, then starts with all various types of say mesoderms and how they really take part in it, right? Neural tube, etc. Meantime, if you really want to know that where is the uterine epithelium, here is the uterine epithelium, right? This is the uterine epithelium. See, the whole thing has embedded and it has gone deep, right? So this one is the uterine epithelium. Okay. Now our focus will move on to the overall picture. That this is the last time we are drawing this uterine epithelium. Uterine epithelium, because now it has completely embedded and it has gone inside. So that's the thing, right? So we have now situation something like this and this, right? So this one would be the amniotic cavity, right? This one would be the yolk sac, and rest is all that cytotrophoblast, etc., right? Which we know, cytotrophoblast, and then the surrounding, etc. This is when it is clearly defined by day 12. From this point, and you remember we call this epiblast and hypoblast, right? This one was epiblast and that's the hypoblast. One change in one day, that is development of, so I'm writing it over here. Say this is like day 13. Everything remains same, but there is development of that connecting stalk. This one, right? So this is day 13, the connecting stalk. Sorry. So we have got epiblast and hypoblast. Surrounding is this cytotrophoblast, right? Cytotrophoblast, and then there is all the say or rest of the cells which are. Meantime, say this portion which is outside, right? That is what? That is chorionic cavity. Correct? Chorionic cavity, right? Properly oriented, right? This is how the embedded things were getting embedded, right? The whole unit when it inside, right? Formation of connecting stalk, the chorionic cavity, yolk sac, and the amniotic cavity, right? From this level, will not be drawing that uterine epithelium, etc. Now we are talking about only one thing, that is the bilaminar disc, right? From this point onwards, bilaminar disc. So we have what do you say? Those columnar cells are called epiblast. Yeah, that's right. They are called as the epiblast. True. Now you have to visualize in a way what we were drawing till this point of time, that was 2D, right? Now you have to think it like 3D. Okay, what if you watch from an angle, right? Because we said that this is like bilaminar disc. Disc matlab to it is one surface and then the second surface. Yeah, exactly. It is like that. It is not just one layer like this. So we are taking the the orientation like this, that this is the disc. It is this. Okay, that's yellow. So let me take the yellow color. So if we watch this particular disc from an angle, from this angle, as if we are watching it from the top, right? So we'll find that yes, that's the disc. It's covering a bigger area, right? And in this, there would be all those columnar cells, right? There'll be columnar cells, right? That would be like this. And then there will be another layer, there'll be other layer, other layer. So basically, it is a flat area where the disk is, and just underneath that disk there would be disk another disk. This will be underneath it, right? Correct. What is there on the top of it? Well, over here there would be amniotic cavity, right? And just below this would be like what? Yolk sac, and this above would be the amniotic cavity. So that's the hypoplast. That's the hypoplast, and this one would be the epiblast. See, in embryology, what happens that you need to understand embryology only once very properly, and don't get lost, right? So because now we'll be entering into these layers. So but every time keep in mind that where exactly are you, because today things would be easier, right? We'll be moving into 2D and 3D, but tomorrow it would be pure 3D, because we have to really understand that how the folding occurs, because then there'll be formation of various parts of the brain, right? There'll be few signaling structures. So these cells, they were called what? Amniotic cells, right? Am I right? Just amniotic cavity and amniotic cells, right? These cells, and then will be forming this entire cavity. So all these amniotic cells. So far, so good. Let's add something to this, because this is where the fun starts. Here is the first stage. We are on day 14 or day 15, right? Roughly day 14 or day 15. And this is the stage of what is called as the gastrulation. Till this point, things were very easy. Now you have to visualize, right? So I'll try to draw as many figures as possible. So this is gastrulation. What gastrulation is? Gastrulation is, say, before giving definition, let's say this is what? This is nothing but our epiblast. Okay, we are drawing epiblast with yellow color. Okay, so let's draw it with yellow color. Just epiblast, forget about rest of the structure, right? So this is epiblast, and we are keeping epiblast simple, but otherwise, so it is like all those cells, right? How the way we drew it up. So this is our epiblast, full good plate, full good plate. Now this epiblast, which side is head end and which side is tail end, no one knows. No one knows. Till this point of time, it is not possible to figure out that which side is head or which side is tail, or which side is the cranial end and which side is the caudal end. The first indication of what really occurs, that this is the plate, right? This is the plate only in the lower half. Only in the lower half. Lower half means the caudal side, right? Caudal side, there is development of what is called as the primitive streak. Primitive streak. Streak means line. So these are the cells, they will start differentiating, right? These are the cells which will start differentiating. Now see, this stage is so important. This day 14, day 15. Female, right? Taking, say, tobacco, right? Any of those, right? Say tobacco, alcohol, right? Alcohol, smoking, right? If the female is diabetic, right? If the female is diabetic, or if there is any radiation, any sort of infection, right? All these insults, then many type of drugs, right? They vitamin deficiency, right? Vitamin deficiency, right? Improper food, it affects this gastrulation. And when the gastrulation is affected, you will see the importance of it. So remember, gastrulation is what? Gastrulation is development of, right? Development of primitive streak. Remember this word, primitive, primitive streak. Importance of primitive streak. One, it develops at the caudal half. Caudal half. So it will indicate that which side is up, which side is down, which side is dorsal, which side is ventral. All those things, they are from this point. Okay? So let's draw this, and then there is development of this, right? This is what is called as primitive streak. Don't worry, we'll be going in more detail, right? And this, the head side, right? Because if this is the primitive streak, this is the head side, right? That head side is called primitive node, and then there is a khadda in between, that is called as primitive pit. So in that is called as pit, and this prominent portion is called as the node. So that is called as the primitive, that means in a very initial stage, primitive pit and the primitive node. And then all the way it will go down, that is called as the primitive streak. Right? Things to know interestingly over here, that would be the cloacal end or the tail end, right? So it is, this is the cloacal end, or the, you can say, cloacal membrane, because that anterior portion, it would develop into buccopharyngeal membrane. So that is where the mouth would develop, and this cloacal membrane, that is where the tail end will develop, right? So this is about, remember, this is about epiblast. Definitely we have got underneath over here, right? All the way we have got what? That hypoblast, right? Hypoblast, just for the orientation. Over here, what we have got, that would be the yolk sac, correct? That would be the yolk sac. And above this entire thing is happening in the base of what? Amniotic cavity, right? Amniotic cavity. Fine, you are with me. But our focus is right now only on the epiblast. Now let's go into still more detail, that what exactly really happens in that. In order to understand this thing, we'll try to watch it from the side, right? We'll try to watch it from the side. So as if we are watching from this angle, right? So we have rotated the whole thing. You will understand immediately. Yellow, right? So they are columnar. So let's draw it like this. So they are all those columnar. Okay? They are like this. But because something developed, right? It was this a prominence. That is the primitive that is the primitive node, and this entire thing was the primitive streak. So it is coming like this, and below below is the hypoblast, right? These are all normal cells, right? Nothing has happened till this point of time, so they are normal cells. Let's fast. So this is hypoblast, and this one is epiblast. So when we look from the side, we come to know that, okay, this primitive streak is above, right? And then this is the streak. Streak means line, and this is the node, which is prominent. And obviously, this cavity is the amniotic cavity. Amniotic cavity. And this one below is our yolk sac. ठीक? yolk sac. So hypoblast, epiblast, those cells, right? For the understanding, let's draw it over here on this side only. This is our visualization from the top, right? Watch it from the top. If we watch it from the top, where we are watching only epiblast, so I should have drawn it in yellow color. So we are watching only the epiblast. In epiblast, we talked about that it has to be on the caudal half. So yeah, this is the caudal half, right? That's the caudal half. So that caudal half is over here. That one would be the primitive node. So I'm just writing primitive node, and this one would be the primitive streak, right? This one would be the primitive streak. This end would be the cloacal end, cloacal end. And how we are watching this, as if we have taken a section, as if we have cut the whole thing, right? At this level, at this level, we've taken a section. So this is the epiblast, right? This one is the epiblast. This one is the epiblast, which we are watching from the top view. This is top view, right? Top view, right? Top view. And this one is the side view, side view. So from the top view, everything is like epiblast, say they have covered. We won't be able to watch what is going underneath. Sorry. What is going underneath. But when we watch from the side and we have taken the section, we find that there are cells which are migrating from epiblast and they go underneath, right? So there is epiblast and then they go underneath it. So basically they are going between epiblast and the hypoblast, and then they start moving. These are the cells which are this, right? These are the cells. These cells, they differentiate and then they are moving like this and like this, right? These are the cells. These are the cells what are called as the mesodermal cells. These are mesodermal cells. We'll be going into more detail of that because these are the cells in which we are very much interested, right? Because they have started forming what's called as the three layer, right? Three layer disc. Okay, so these are mesodermal cells, right? Achha. If you go slightly in more detail, right? Let's make space. You know that this is yolk sac. Think it like that this is what? This is primitive node. This one is streak, right? And now think it like we are watching from the top, but the cells are going underneath, right? Cells are going underneath. So there'll be few cells. There'll be few cells which are from this node. So this is the cranial end. This is the caudal end, right? This is the cranial end. So from the node which is at the cranial end, there would be few cells which will be going like this, right? They are paraaxial mesoderm, right? Paraaxial mesoderm. Paraaxial, that means this is the long axis and surrounding the long axis. Then they'll be at the caudal end. Are you getting it? Right? So this is, if this is the whole surface, this is the whole surface. That on your side, it is the cranial end. So cells which are going from there, right? They are paraaxial. Then there are cells which are on this side, that is at the caudal end. When they are going, so they will be going like this. So that is called as the lateral mesoderm. Lateral mesoderm, right? And then in between, that's the cranial end, this is the caudal end. So caudal end cells which are going underneath, right? The caudal mesoderms which are going between epiblast and hypoblast, they are lateral mesoderm. All these things, lateral mesoderm, paraxial mesoderm, they'll be forming amazing structures, right? So, and then there would be in between. So very conveniently we just tell them intermediate mesoderm. So when we'll start discussing about that, let's discuss about the formation of dorsal ganglia, right? Dorsal ganglia. So at that point, when we'll be talking about the terms like paraxial mesoderm or intermediate mesoderm or the lateral plate mesoderm, there should not be any confusion, because everything is in response or in relation to primitive, primitive node and primitive streak, right? This is the streak. The moment we say that we are talking about lateral mesoderm, lateral plate mesoderm, that means that is occurring at the caudal end, right? When we talk about the paraxial mesoderm, that means we are going towards the cranial end of the primitive streak, or we are near the primitive node. Crystal clear, right? Now on to the next level. So as they go, so at any point of time, now we have got, again, if we really draw this thing, right, if we really draw, so this is one layer, right, then green layer was our mesoderm layer, and this one is our endoderm, right? So now we are in position to answer that all these layers, right, they are now, previously till this point of time we were talking about that they are hypoblast and epiblast. Now we'll start telling them with the formation of these three, we'll tell them ectoderm, endoderm, and in between is the mesoderm. So here it is, this is now ectoderm, right? Question, what's the question? So the primitive streak is like a base for those mesodermal differentiation. Yeah, exactly. But say primitive streak is the point. Primitive streak is the point which will be giving some more important things also, right? Just within 2 minutes we'll come to that, because that's the next step. But yeah, this is primitive streak is like a reference point, that when we say lateral plate mesoderm, so then we keep on confusing a lateral plate, matlab where exactly are the lateral plate? Well, lateral plate is when it is at a caudal end of primitive streak, right? So remember primitive streak as a is like a base, or yeah, or like a reference. Fine, right? Okay, so that is ectoderm, and we have this one is mesoderm, mesoderm, and this lower one, that would be the endoderm. So we have ectoderm, endoderm, and mesoderm. Now we have got all the right to call this thing as three-layer trigeminal disc, right? So this is trilaminar, that is three-layer germinal or germ disc. So we have got three layers, right? Yes, this portion, that would be the yolk sac, just for the reference, right? And yes, this one would be the amniotic cavity, amniotic cavity, or the amniotic sac. Now we are entering into only this small portion in this, right? So we are entering into the mesoderm. This one is lateral mesoderm, right? Which one? This one? This is this what we have drawn over here, right? Well, from which layer we cut, right? Let's see this. Meantime, in case if you are curious that what exactly is formed with these, so let's say we write it like ecto, meso, it will change the color. So ecto, endo, and the meso. So epiblast form ectoderm. That's right. That's right. And see these epiblasts, now they are ectoderm, right? Then there will be someone coming and it will say, कि चलो यार, you become neural cells. So then that ectoderm will change the terminology to neuroectoderm, right? So it is like that you have got various gundas, right? But some gundas, they are expert in say fighting, free hand fighting. Some gundas, they are expert in say knife. Some gundas, they are expert in guns. So this is what is called as differentiation, right? All of them initially they are same, but then they differentiate into one specific quality. So that ectoderm with a specific signal, they will become neuroectoderm. So they'll say, okay, okay, we'll take the responsibility of formation of brain and the formation of spinal cord, right? Leave that thing to us, and they will all the way they will start their work. So thus, all various different types of that is gene differentiation. Yeah. Okay. So chalo. Ectoderm, what it will do? Ectoderm will make CNS, right? You know what is CNS, right? Brain, spinal cord. Ectoderm will make peripheral nervous system, right? Peripheral nervous system, right? Important points. It will make retina, because retina is considered as extension of brain only, right? It will make cornea. It will make lens. So now we know that this is so important, right? Sclera and ecto, outside skin. See? What about the meso? Middle layers. Everything in the form of, right? Say this muscles, cartilage, right? Cartilage, connective tissues, right? Connective tissues. All they are thanks to this. Then, yeah, obviously bones, right? Bones. Then it will make very important structure, and that is heart and major vessels, right? Heart and major vessels. That would be by him. Then kidney, ureter, gonads, right? All are kidney, ureter. Then kidneys, so then obviously the cap of it, that is adrenal cortex, right? So adrenals, that is all by mesoderm. Endoderm, endoderm, they are super specialist. See, the all the path, GIT. So tonsil, starting from the tonsil, then thyroid, right? Then as you go, this is thyroid, obviously parathyroids, right? Parathyroid, then trachea, right? Trachea, thymus, right? Thymus, then GIT, right? GIT, then liver, lungs, pancreas. So basically, it is over there. And as you go down, the bladder, the urethra, that is by endoderm, right? Over here, the gonads, they will also be the by mesoderm. So that's the importance of these trigeminal. Lymphocytes and rest of the organs. See, basically, depending upon either their mesoderm and which organs are we talking about. Anyway, it will come, right? We'll be discussing it in still more detail, right? This is just for the basic understanding. Don't worry about rest of the organ. Our focus currently CNS, right? Now moving on to the that important section where we want to see the first vital structure, which is like a signaling structure. What exactly we mean by signaling means, say just now you asked, right, that is it like, say when we say that this ectoderm, right, ectoderm will be telling about that let's form that neuroectoderm, right? But who tells that? The first structure which is developing, and the first structure which we need to know, and this is pretty confusing, right? It's pretty confusing, so we'll try to draw it in this way. This and this, right? So this one below, this is endoderm, and this one is ectoderm. Okay. So endoderm and ectoderm. Yeah, what's the question? At what stage any defect can be detected? Very simple. Whenever things are developing, right, there will be various types of defects occurring at various levels. Right? So it can be detected only at point when it is developing. So we'll talk about it, right? Just because the real defects, they will start from this point onwards. Because otherwise, if any defect is occurring at this level, right? Over here, that is the three germinal layer or anything. If it is happening at say primitive streak, etc., so that will lead to massive, at what stage? Huh? It will lead to massive changes, right? Because if these cells, if this streak itself is not developed properly, so then the chances are, because this streak is something which will be giving rise to so many important structures and landmarks. So that's why the biggest insult of this say day 14, day 15, tobacco, alcohol, smoking, etc., right, they are so injurious. First trimester, never ever take the X-ray, because that thing can lead to devastating changes, right? Huh. So here it is, the simplest explanation, ectoderm and endoderm. And in ectoderm, we said that there would be formation of what? There would be formation of that primitive streak, right? So initially primitive node and the primitive pit. So here is, let's draw this thing, right? Let's draw this is as primitive node, right? So, so we have got now situation like this. So this is primitive pit, and this one is the prominence, the primitive node. Okay, so you know this part. And obviously this is ectoderm. There would be formation of cloacal membrane. So this is cloacal membrane, right? On the, so the moment this is happened, right? So this is, if I draw it like this becomes the cranial end, है ना? This is the cranial end, because primitive streak, which will be this, this is the primitive streak. Primitive streak develops in the caudal half, right? Primitive streak in caudal half, caudal half. So that cloaca, that opening, anal opening, that is defined. So here comes now the first one over here. This is the buccopharyngeal membrane. So that is the opening of the mouth. So mouth would be formed over here. So that's the mouth opening, which is called as the buccopharyngeal membrane. Right? Buccopharyngeal membrane. Here, as we said, that there will be the formation of mesoderm, right, which will be going in between. It starts from here. From this primitive pit, right, inside. Now I'm just removing this, right, for the clarity. There is a development of few cells and they will start traveling cranially. They'll start traveling cranially, right? When these cells, they develop, right, and they keep on moving. They keep on moving till what point? See over here, over here. Now we draw it like this. Over here. This is what is called as the precaudal plate. So this endoderm, it also forms a depression, and that is what is called as the precaudal plate. So this is the point. This is the point where there will be mesoderms, and this extension will come all the way till this point. Will come all the way till this point. Okay? This is what is called as the notochordal process. This is notochordal process. So it extends all the way till precaudal. Getting confused? Should not. Again, see, keep telling me if you have understood. All right, I'm repeating all the points. Buccopharyngeal membrane clear, right? If buccopharyngeal membrane clear, so then just underneath the endoderm, that leads to formation of precaudal plate, just another depression, right? So this is clear. The posterior portion of precaudal plate, it leads to few of the cells, and they are called as the precaudal mesoderm. So these cells, they are precaudal mesoderm, right? And these are the mesoderm cells, because we just said na, that they will be going underneath. They will be going underneath between ectoderm and endoderm. So this entire portion, which is from the caudal half, is going towards the cranial part, right? It is going towards the cranial part, right? And it will go all the way till this, see, almost to the mouth. That's the mouth, right? This is which is what is happening between both them. Is the streak place? Streak is here. Streak is here. Why are you confusing? Streak is here. Streak is what I said, that it is into the caudal half, right? This is the caudal half. This is anterior, cranial, right? That's what I said, cranial end. In cranial end, so there is no streak, right? And this is the cloacal half. So that is this portion, that is the primitive streak. We are not talking about the primitive streak. We are going into the reverse direction of primitive streak. This is the notochordal process which is developing from primitive pit between the two layers, and it is going anteriorly and reaching to the mouth, right? Reaching to the mouth. This is notochordal process. Got it? Then starts the canalization. Canalization means the cells which are, right, the cells which are in between. So it was not placed vertically but horizontally in the plane of ectoderm. Oh, obviously, right? That's what I said. We are watching from the side. We are watching from the side because see, this is the top. Ectoderm is on the top. Endoderm is below, right? It is exactly this plate. This is what we are watching. This one, right? But taking a cut section, right? So that is anterior. That one is posterior, cranial end. Let's make it still better, but we'll draw less, right? So yeah, so here, here, right, and this, and the new development, it has become like this, and there is formation of a canal, right? There is formation of canal. The cells which are into the center, they go for apoptosis, they die, and then there is formation of this canal. So this notochordal process, which was pretty steady, right, which was pretty strong, right, now it is leading to the development of canal. Now comes very interesting thing. You have to orient yourself to understand the best part of it, right? Say this is the situation, right? So this is what this will be, future mouth. So this is basically buccopharyngeal membrane, and this one is like that pit, right? You know this. This is the pit, right? Primitive pit. This would be the prominence. That would be the primitive node. This would be the primitive streak. This one would be the cloacal end. And if I ask you, where is the amnion or the amniotic cavity, that would be over here, right? So this one would be the amnion, and this below part, right, that one would be the yolk sac. Good, right? Very clear. And yes, the layers, this one would be the mesoderm, but the difference this time what we have talked about is, I'll draw it again, the mesoderm, as the canalization occurs, right, something different happens. This, right, this goes like this. See the difference, it goes like this, So have you noticed one thing? That as the canalization occurs, right, this notochord, right, this notochordal process is going towards endoderm, right? It is going towards endoderm. See the difference over here, right? This is ectoderm, this is endoderm. As the canalization occurs, this whole unit, right? It starts coming down, right? Very important thing. It starts coming down. So it has developed this canal, right? And it is coming towards the endoderm, right? Towards endoderm. We are about to watch one very classical thing, what is called as the neuranteric canal, right? There is lots of confusion that where exactly is this neuranteric canal. What neuranteric canal is, see, as this develops, right, as this develops, now the lower portion, right, see how interesting. I have to really draw it again, right? Okay, let me copy this page, duplicate page. So we have got this, and then see the next change, right? In this change, the endoderm, now it goes like this and this. So the endoderm which was over here, that is taken off, right? That is degenerated. When this is degenerated, this is like there is a formation. That pit is already there. That pit is open. Right? So it will lead to this canal. This canal. I'll remove this endoderm written over here. This will lead to this canal. So this is a canal which is between amniotic cavity and the yolk sac, and this canal is called as the neuranteric canal. Neuranteric canal. No doubt this is temporary, after that it gets closed, but this neuranteric canal is like a temporary connection between amniotic cavity and the yolk sac, right? Very, very typical thing to understand. When it occurs when the notochordal process, right? Notochordal process, it goes down. It goes down, and as it touches the right? Endoderm, as it touches the endoderm, so the portion of that endoderm that is taken off, right? Once it is taken off, so then it leads to formation of a connection between amniotic cavity and the yolk sac, and that is what is called as the neuranteric canal. Neuranteric canal, right, which is temporary connection. It is the temporary connection between yolk sac and amnion, right? Well, there are various theories that what is the importance of this neuranteric canal. One of the theories that it has got a very specific role into the exchange of substances between yolk sac and the amnion, right? That is the role in exchange of substances. One of the another theory, it also tells that it is to balance the pressure, right? To balance the pressure between two chambers, to balance the pressure. But whatever, this is the concept of neuranteric. Now to make it still better, right, let's see it in this format, right? This thing will make your concept super easy. So here it is. Let's try it a bit smaller. Oh, wait. Yeah. Okay. This and this. So we have got ectoderm and endoderm, right? This is ectoderm and this is endoderm. And in between, we have got that notochord, right? Notochordal process, right? Notochordal process. and canalization has started, right? So that's why I've drawn it like this, that canalization has started. It goes down, right? So it goes down. So the next would be something like this, that it it has reached over here, right? It has reached over here, and it's something like this, correct? Fine. So this is where the neurontric canal will be prepared, right? It will come into picture because this endoderm has been taken off. Now it will take away. The next stage would be this. Next stage would be this. And this is, till this point it was called as the notochordal process. Now this is called as the notochordal plate. This is called as the notochordal plate, right? So it came down all the way, and this is how it is formed, right? And the next stage would be, this plate would proliferate, and the next stage would be this. It will go back. It will go back, right? So here, say this is notochordal process, right? Don't confuse. This is the process. In process, there is canalization. So when the canalization starts, it starts going towards endoderm, right? When it reaches to the endoderm gone, right? That much portion. And then the portion of this notochordal process, right? That also goes. It leads to that formation. The new name is notochordal plate. So this is the plate which is formed. And then it proliferates, and then it starts the return journey, right? When it starts going back, now this is like a solid rod, right? A solid rod, and this rod is called as the notochord. So in figure, when it is said that, okay, this is ectoderm, this is endoderm, and in between we just draw this notochord, actually notochord goes into this formation. This is why it goes like this. And what's the importance of it? Well, its importance is nothing develops from it. Nothing. Because this is going to get degenerated finally. And it will end up in what? Nucleus pulposus. Right? This is the final fate. Nucleus pulposus. You know what is nucleus pulposus, right? So this is one vertebra, and there is intervertebral disc, and this is another vertebra. So it is this, right? If you just take out this one vertebra, so then it is this center one, because over here these are all annular fibers, right? Annulus fibrosus. So this in between, that is the nucleus pulposus. That nucleus pulposus, it tells that once upon a time, all the nucleus pulposus, all of us were called as the notochord, right? And the importance of this notochord is super importance. They are the midline signaling structure, because the entire brain is formed by the neural tubes and then the somites, right? Neural tube, and then it will lead to formation of brain, spinal cord, all those nerves. But the main signaling, that is by the notochord. So this is the chief midline signaling structure. It is this structure, once it is formed, it will start giving the information, sending the signals to the ectoderm, that okay, from this point onwards you'll be called as the neuroectoderm, and the process starts. And that process, which leads to formation of neural tube and somites, right? Neural tube and somites. We'll just in a minute we'll start with what is called as the neural tube. But this is what is called as the notochord, right? So till this point, again, the top portion, that is where the amnion, and the lower, that is where the yolk sac, right? So I hope you are properly oriented that what exactly are we trying to talk about. ठीक है? And this is nucleus pulposus. ठीक है? Let it remain like that. One more word which you should know. The thing what is called as the neuropore. Pore means opening. Pore means opening. ठीक? So how awesome would it be if now we see the whole assembly in a way as if we are like a car repairer, and we go underneath the car and then we watch. So what I'm telling is we watch this thing from below, right? We watch it from the below. That is from the endo endoderm, right? From the endoderm perspective, we watch it from below. Sorry. Then concept of neuropore becomes very easy. So when we are watching from below, I'll take just one color. So there would be one top end. This is called as the cranial neuropore, right? Cranial neuropore, and all the way it developed, and then there would be the caudal neuropore, right? Caudal neuropore. There would be development of many somites. They will be developing in between, right? And then obviously this is the lower end, so we'll watch that yolk sac. We'll watch the yolk sac. Right? So this is the scenario when the neural tube will come into picture, right? So roughly about day 22, 23, right? This is where the neural tube, this we are introducing a new character, neural tube. Right? So where is this, how it is developing? Starting with back to that structure. So where now we'll be drawing it like this. One, then two. Our notochord is here, so we'll draw it is in the form of a solid structure, and surrounding it, this green portion, that is mesoderm, right? Right. So this is how we'll keep it, right? Ecto, meso, endo, and notochord. Bus. From this point onwards, we'll not label. Right? This process is called as the process which will be leading to the formation of neural tube. This is called as the neurulation, right? Neurulation. What neurulation is? One simple thing. Notochord said, right? Notochord has sent the signal. Notochord signaled, and neural tube formed. Neural tube formation. So our next step is how the neural tube is formed, because once we know neural tube, it is easy, because brain spinal cord will be coming from that. Right? Notochord will tell whom? Notochord can tell only ectoderm, because that's the area from where the things will be. So neural tube formation, that would be from ectoderm, right? And because it is by the notochord had sent the signal, so we'll now name it as neuroectoderm. Neuroectoderm. So ectoderm is like a storage. It has got cells, right? It will say send few cells ke jao, you all become neuroectoderm. So they will go, they will isolate themselves, and once again the ectoderm will keep its layer as intact. And those cells which have gone inside, they will become the neural tube, and later on they will be leading to the formation of brain and spinal cord, right? So basically from ectoderm they are released, then they are cut, they are isolated, and they said you do your business by yourself and you become neuro, right? While ectoderm will once again make that layer intact. So how it forms, this is the whole structure, or this is the whole concept. So first one, right? That's the notochord, right? I'm keeping just one color. Whenever needed, I'll change it, right? And the endo, right? So this is ecto, endo, and obviously over here the meso, and this one is notochord. Signal arrived, right? So there were changes in the ectoderm. So ectoderm will go like this, and it will lead to midline thickness. This is thickened. Midline thickness. So that midline thickness, this is the midline thickness. So obviously cells proliferate, right? And it will start going down, right? This midline thickness is called as the neural plate, right? It is called as the neural plate, right? So basically it is the neural plate, or the, from this point onwards you can say it neuroectoderm, right? Neuroectoderm. So it will be having the cranial end and the, because it is happening at full length, but say, and below over here it is the notochord, right? There are mesoderm and everyone is there. Still it goes down, right? So this is step 1, this is step 2, now comes the step 3, right? It goes like this, still goes deeper. So where is our notochord? Here is the notochord, right? Where is our endoderm? All are there only, right? Mesoderm, yes, that's the mesoderm, right? Theek. All those mesoderms which will be making all those muscles, cartilage. So things are, this process is called as the involution, right? This is because it is going down, right? So it is called as the involution. More than process, we are interested in these areas, right? This is what is called as the neural groove. So makes sense. I'm giving it the name neural groove. But we have got very specific interest on this area. On this area. The areas which are just unfolding and then the folding and they are going inside, right? This. So that is what is called as the neural fold, right? It is called as the neural fold. This becomes more and more thicker, and now we'll call that thing as neural crest, right? Neural crest means what? The cells differentiate. Cells start differentiating, right? These cells, they will become very special cells because they will be isolated, right? So how it occurs? It goes like this. It goes like this. So this was the edge. This edge was like two waves, right? And those two waves, they are meeting, right? Those two waves, they'll keep on getting, say previously the fold is like this, then it becomes like this, then it becomes these two areas, they start coming near each other like this, right? This one, then it will be like this, and then it will be isolated, right? So here it is like this. Our notochord is over here, rest all structures same, right? And the finally it becomes this. Here is very proper tube, because see, this is a blank space, right? This is a blank space. So when only the two tubes they meet over here, so in between, so there will be space. and just below it, this is the notochord, right? There would be all the mesoderms over here, mesoderms over here, and our endoderm over here, right? So we have formed neural tube. This one notochord, this one mesoderm, this is endoderm, this one ectoderm, right? Above we have got amniotic cavity, below we have got yolk sac. Done, right? Acha. So if we differentiate, right, notochord was its purpose signaling, right? In one word, signaling, signaling to that ectoderm, right? Then there was neural plate, right? Neural plate, which was occurring on about say 18th day, right? 18th day, day 18. So it was like this, right? This, and the neural plate was developed. So when it started becoming thick, right? Then the neural tube, right? The neural tube. So when it became like this tube, and over here it was the notochord, right? There was all mesoderms and the endoderm, right? So this neural tube is what, as we said, this is technically speaking embryonic precursor, embryonic precursor of brain and spinal cord, that is CNS. So if it is embryonic precursor of brain and spinal cord, it has to be divided, right? It has to be divided into, say, all the parts. Now see, it is this point. In case if there is any anomaly into the brain, right, into this neural tube, so then it can lead to anencephaly. Or if anything goes wrong into the region of spinal cord, so it can lead to spina bifida, right? Spina bifida. We'll see even the cases of these, but right now, let's see that neural tube, right? This is the neural tube. This neural tube would be having four sections, right? Four sections, very easy sections. And it has to be like that because when that tube is formed, then this tube will start folding, and this tube will be what at cranial end it would be thicker. As it will go back, it will become thinner and thinner. Makes sense, because on the cranial side, say this is forebrain, right? Then there would be midbrain, then there would be hindbrain, and the spinal cord. So these will be the areas. Right now, neural tube is just like a tube. Only thing that at the cranial end it is thicker. Bus. So this forebrain, right? It is also called as the prosencephalon. Prosencephalon. Midbrain is also called as the mesencephalon. Mesencephalon. Then hindbrain is called as rhombencephalon. Rhombencephalon. And spinal cord is spinal cord, right? Spinal cord is spinal cord. So these are the, then, so that means this neural tube, you can guess that neural tube is straight, then it will start dividing, right? One part, two part, three part, and then going like this, right? Yeah, exactly. This is what really happens, right? Because that's how it will lead to formation of all this. What about those neural crest cells? To make it still more clear, right? I'm drawing one image. This is what ectoderm. Then this is, yes, you are right, that is neural tube, right? Neural tube. Then this one is notochord. You are right, notochord. And then in between, right, this is, these are the cells which are neural crest cells, right? These are neural crest cells. From where have they come? See, you remember we were telling about this, that this is what, this is neural, we have already written, this is neural plate. So those neural crest cells, they are this and they are this. So from here they migrate laterally, right? Because in any case, finally, the ectoderm is going to cover it, right? So those crest cells, they go like this, separated from the neural plate. So the whole process, it goes like this. This is the ectoderm, right? There is involution. So, but these cells, they are the neural crest cells. As they unite, the tube, it goes below, but those neural crest cells, they go sideways, right? And they form a layer. Notochord is obviously sitting back, and this would be the situation. These are the neural crest cells, right? So what are these neural crest cells? These are the new cell types, and they neural tube and they migrate, right? Remember, they leave neural tube, right? They were they were sitting on the corner, right? Sitting on the corner, sitting on the edges, कि बस ये neural tube बने अपने भागे, right? Ectoderm से भी अलग, neural tube से भी अलग, and that's how they move, right? So leave neural tube and migrate, and within a minute you'll understand that yes, it has to be like that. The reason behind it is that if neural tube is making central nervous system, there has to be someone who should make peripheral nervous system, right? Thanks to neural crest cells, they will be making peripheral nervous system, right? Because finally to it is neuroectoderm. Notochord said, I'll speak only once, right? Neuroectoderms come. So then they said, this is the package of neuroectoderm. Few cells, they'll make neural tube. So then it is the business of neural tube to make brain and spinal cord. And what neural crest, they are the some cells isolated, will go on the periphery and will make that peripheral skeletal system, right? So peripheral nervous system. So that's how that would be formed. Interestingly, it also forms the face skeletal system, part of it, skeletal system, right? That is also formed. And because these are the neural crest, and there one more function, that is the melanocytes. They will also make the melanocytes. Melanocytes are skin pigmentation, right? Which gives color to your skin pigmentations. So brain, spinal cord strictly, that is by the neural, and that's how it leads to formation of, say, this entire system. Last, just for before we close for today, right? Because from tomorrow we'll be starting with the rotation, bending, and the formation of every part of the brain. One last thing, from the neural tube perspective, right? From the neural tube which is formed. So you saw that initially it is open, right? Initially it is open, right? Over here it is, right? This tube is open, this is open, and then it starts getting closed. So if we see it, right, this is one important piece of information. If we see that this is the cranial end, this is the caudal end, right? Caudal end, cranial end, it closes on 24th day, right? And this closes on 26th day. So typically it's like that zipper, right? Zip. So just from the top, it takes down and it starts getting close. So previously, if we really draw it like that, okay, these are the neural folds, right? And in the into the base there would be the neural plate, and then those folds they meet, right? When they those folds they meet, so they will be meeting like this, but this lower portion is still open. So as the time passes, right, this is closed and this also gets closed. Still the lower portion is open. And then finally over here, this is closed, and it like that. So it takes 2 days for the zip all the way from the top to bottom and close everything. And that is where in between starts developing the what is called as the somites. Somites means all those sections, right? There'll be occipital, cervical, thoracic, all those areas, right? Because that is after fused neural folds. When the neural folds they fuse, so final output will be there would be ectoderm, then there'll be neural crest, right? Neural crest, those spatial cells which will be making peripheral nervous system. Then there'll be neural tube, and then just still underneath is notochord, and finally the endoderm. So we this would be our final stage, right? So we stop at this point. I just saved this thing. We stop at this point. Tomorrow we'll be starting our journey from this point onwards, right? So thanks a lot. Thank you so much. We are putting more emphasis on embryology because once you understand this now, so many congenital defects and so many parts of the brains, everything becomes very easy, right? So thanks a lot, and see you tomorrow for the second part of embryology. Take care and see you. Bye bye.